to you about some preclinical, cannot hear me? Some preclinical data about combination of JAK and HSP90 inhibitors in the treatment of myelofibrosis. So our lab studies myeloproliferative neoplasms, and these are blood disorders which are characterized by increased production of one or more type of blood cell. So for example, in the case of red blood cells, this disease is called polycythemia vera. If there's an increase in platelets in the patient sample, this disease is called essential thrombocytosis, and then there is primary myelofibrosis. So MPN patients actually present with a lot of symptoms, including bleeding, thrombosis, splenomegaly, and also a lot of bone marrow scarring. Really, the pathogenic cause of this disease was unknown until 2005, when several groups discovered a mutation in a gene called JAK2, or in the upstream receptor called as MPL, and both these genes actually both these mutations actually cause an activated JAK-STAT signaling pathway, which can now be targeted using the JAK inhibitors. So how effective really are these JAK inhibitors? We know that patients feel great with JAK inhibitors, and the most advanced of these JAK inhibitors is ruxolitinib, which is Incyte 18424 from Novartis. Patients show improvements in splenomegaly, constitutional symptoms, and there is actually reduced progression to leukemia. However, in a majority of these patient samples, we do not really see a decrease in the mutant allele burden, which means that these inhibitors are unable to cure the patients, unlike imatinib with CML. So we also see this in our mouse models of disease. So here in this graph, I'm showing you the percentage of GFP, which is a marker of the disease in these mouse models. And we see here, even with the end of treatment, you do not really see a difference in the GFP levels, suggesting that the inhibitor is unable to cure the disease. So we try to model this in our cell lines by growing cells which were naive to begin with to JAK inhibitors to increasing concentrations of Incyte 18424. And yet, at the end of about 4 to 6 weeks, we could generate persistent cell lines which were growing in high concentrations of the inhibitor. What we found was the most seminal observation in these samples was that only in the persistent cell lines, we saw this association of these other JAK family members, namely JAK1 and TYK2, with phosphorylated JAK2, and this could not be seen in the naive samples. So this suggested to us that the other JAK family members, which normally do not associate with JAK2, are now associating with phosphorylating JAK2 in these samples, and therefore they restore the JAK-STAT signaling pathway and thereby make these cells insensitive to JAK inhibitors. Now, because the JAK-STAT pathway was still active, we wanted to know whether these cells are still JAK2 dependent. So to answer that question, we took JAK2 hairpins and tried to knock down the levels of JAK2 in these persistent cells. And here, in terms of cell growth as well as signaling, we see that the naive cells, not surprisingly, but also the Incyte persistent cells were dependent upon JAK2, and they were dying when we knocked down JAK2 in these cells. So this suggested to us that instead of inhibiting the kinase activity, we could try to, if we could selectively degrade JAK2 in these cells, we could get better efficacy. So about a couple of years ago, we published a paper using an HSP90 inhibitor called PUH71, which shows very good efficacy in mouse models. So this is a survival plot showing that mice that were treated with PUH71 lived throughout the treatment, whereas mice that received a vehicle or a placebo died very soon. And in this plot here, we see that what's surprising is that PUH71 seems to be selectively taken up only by the malignant cells. And here it's getting And only in the malignant cells you see degradation of JAK2 and not in the naive cells, suggesting that there is a therapeutic index for PUH71 in these cell lines and in models. So trying to combine these together, we made a bone marrow transplant trial where we took sick mice and then we randomized them to receive either vehicle or a combination of Insight with PU-H71, and this Insight was a low-dose Insight. We also had an Insight single arm just to so that we could combine what's happening with single therapy versus combination therapy. These mice were treated for 2 weeks, and then we sacrificed a cohort of the mice to look at downstream signaling. For the next 2 weeks, we split the mice that were receiving the low-dose Insight and the PU-H71 to continue to receive the same treatment. In the second arm, we increased the concentration of Insight to a full-dose Insight. For the mice that received Insight alone, we again split these into 2 combinations, one that continued to receive single-dose Insight, and in the other one, we added on PU-H71 to see if the efficacy of addition of PU-H71 could be as efficacious as combination. So these are just data looking at the WBC cells in these mice. The vehicle mice are obviously very sick, but you see that with the combination treatment, you see a very nice decrease in the WBC cells, even in comparison to the mice that received monotherapy. So what happens at the end of treatment? We find that this treatment is still efficacious. Even at 28 days, we could see that combination therapy was much more efficacious compared to single therapy. When we add on PU to Insight monotherapy, we found that this addition was still efficacious, and if we increase the concentration of Insight, this was further efficacious compared to WBCs, compared to the Insight monotherapy. We saw similar efficacy in the reduction of platelets as well as spleen weights in combination therapy in comparison to monotherapy. We also saw a reduction in the JAK-STAT signaling pathway that we did not see with monotherapy in these mice. So in summary, we have shown that JAK2 persistent cells remain dependent upon JAK2 expression, and we could leverage this pharmacologically using specific HSP90 inhibitors. Combination therapy of JAK and HSP90 inhibitors decrease blood counts, platelet counts, as well as spleen weights. And we feel that because of these data, clinical trials for combination therapy are warranted, and these are being planned to improve outcomes in MPN patients. Thank you.